Okay, now for something completely different. We're going to try and continue this discussion about anesthesia. And I'm going to show you some of my work. And by the way, I'm glad to be here, and thank you to the organizers for inviting me. So we're going to look at if anesthesia removes consciousness and if microtubules are involved, can we look at other systems? And in my case, I'm a photobiologist and I study plants. And I wanted to try to see whether plants would respond to anesthetics as animals and humans do. And this is nothing new. Many people before me have tried anesthesia on plants. Many times, they have looked at like the fly traps that will stop moving or they will slow down growth. But I wanted to think of a different mechanism or a method where I could quantify the system. And one of the things that we know that consciousness that we experience is a response to the environment. So one of the things that we do consciously is to respond to our environment. And of course, plants do that as well. And as a photobiologist, I chose light as the environmental signal to see how plants would respond to light, and that can be quantitative. So basically, we are going to talk about that, but I want to broaden our reach a little bit. So we know that consciousness may not be unique to humans. We just discussed maybe other animals, and some people think dogs definitely have some form of consciousness, dolphins, and these are accepted, right? We can also think that maybe it's not even unique to Earth. Well, these days, with all the UAP talk, if there are actual aliens that are landing on the planet and been doing that for many thousands of years or whatever that we keep hearing, then obviously they have some form of consciousness. Even if we don't believe any of that, I think it is pretty difficult to digest that this planet in the entire universe is the only planet with conscious beings. So let's, at the very simple level, let's just accept, okay, it's possible that there is consciousness outside of Earth. So that means if we take that step, that means consciousness did not just evolve on this planet, it possibly evolved elsewhere as well. So if we can take it that far, then I can tell you plants are not that far behind. So I'm not saying that plants are conscious. And because we have no proof of that, particularly the type of consciousness that we experience, cognitive and those. However, if we are able to respond to our environment, we are 100% certain that plants also respond to their environment. And if our ability to respond to the environment can be inhibited or suppressed transiently by anesthetics, can then be true for plants? That's the question we're going to ask today. So if plants can respond to light, and then if we give anesthesia to plants, will they transiently lose that ability and then be able to respond to light afterwards? Okay? So this work was done at Carnegie Institution at Stanford in collaboration with Dr. Bruce McIver. He's Professor Emeritus now from Stanford University Anesthesia. Also, I have to say and acknowledge that I started working on this project because I was thinking about consciousness in general, and plants just happen to be my subjects. But I started to look at microtubules mainly because after I met Stuart almost 3 years ago, and we've been looking at microtubules in plants. So that's what I'm going to show you today. Okay? So basically the question is, this is how a plant grows in the dark, and if the plant does not see light, until it sees light, it will grow like this, which is an elongation growth. We call this etiolation. Etiolation is a word that's coined from etio, means unknown signal. Relation is response to a signal. So etiolation is response to an unknown signal. That's how it grows in the dark. When the plant sees light, it grows, its morphology changes dramatically. Starts to green, This is day and white light cycles, like you will see in a rhythm of like a day and night. And you can see chlorophyll development, and the elongation growth will suppress, and the plant will start to photosynthesize. So we understand this, that early on after germination, the plant is trying to come through the soil. Once it reaches light, it can photoautotroph. It can make its own food, which we can't because we don't have chlorophyll. So plants make food for not only themselves, but all the other living organisms on the planet. So what if now we use the photoreceptors that are involved in response to light in the plant, but we give the plant anesthesia? So one of the very typical responses that we study in plants is called phototropism. Phototropism is characterized by the ability of a seedling, this is about 3-day-old seedling, responding to a light signal which is coming from this direction, from the right-hand side, and it's going to bend towards the light. And this bend is close to 90 degrees. And you can see that, we can measure that. So if there's a seedling, it will bend towards light. And by the way, it only bends to blue light, towards blue color light, not any other color, not red, not far red. Obviously white light, because white light will contain blue light. So this is called phototropism, and we can measure the degree of angle and suppression of the phototropism as we increase isoflurane, which is an anesthetic. So you can see isoflurane concentration, as we increase from no isoflurane to increasing amounts of isoflurane, we lose the curvature. So what this is showing is the hypocaudal curvature, the bending, and you can see that without any isoflurane, it's bending about 90 degrees, and as we increase the amount of isoflurane, we lose the bending almost to straight, no phototropism. Now this occurs within 3 to 5 hours. Okay? Now what happens if we let it sit under this condition, these seedlings? We leave them under these conditions for 10 hours, so that's another 4 to 5 hours after the anesthesia was administered, which was only 30-minute application before the experiment started. So if we leave them for another 5 hours, we can start to see recovery. So these seedlings will start to bend at certain concentrations of anesthetic, and obviously when we reach a higher level, a higher amount, then we don't see recovery, which is also true for animals and humans. So we can see that by giving isoflurane at the, I would say this is like the sweet spot, which is 0.075%, that plants have just experienced anesthesia, and they will stop responding to the photo signal, and then after about 5 to 6 hours, they are able to respond again. So I'm going to show you videos of this. Are you ready? You can see plants responding to anesthesia. So what we have here is a corn plant. This is a control. Then we can see increasing amounts of isoflurane being given, and then these plants were put together in the same, under the same light condition and imaged. And this is 0.3%. So if you observe this one, this time lapse is about 8 hours. So you will see that in the first 3 to 5 hours, you will see these seedlings responding, these responding slightly less. And then after about 5 hours, this will start to move. Okay? Oops. Have to play the video, please. It's at the bottom of the... So you can see these are moving, and then 5 hours later, they start to move. So this is corn. Let's go to the next one. So in this case, we have pea plants. These two are control, and these two are 0.3%. So can you play that one, please? So you'll see that in this case, this particular plant really loves light. You're going to see how much it will respond to light, and these two will not respond for quite a while. Can we play the video, please? Okay, so because this goes for 11 hours, you will see how this is tracking light, and it's almost going to respond in a way that is bowing towards the light signal. So again, this is 11 hours. So it's going to move, and so it's showing a little bit rhythmic diurnal movement there, and then slowly it's going to go straight towards the blue light. You can see this is starting to move, this is starting to move, and this one has not moved at all yet. So we can see that by giving anesthesia, we can block the ability of a plant to respond to light. Is that consciousness? That's at least an ability to respond to an environmental signal. But what is underlying this mechanism? Let's go to the next level. So we created fusion protein with a green fluorescent protein fused to tubulin, which tubulins, as we know, make microtubules. So this is a GFP tubulin, which we can observe under a confocal microscope. So if you look at a wild type, which means, compared to a mutant, in this case we are using COP. COP stands for constitutive photomorphogenic. It's a very well-studied mutant. But under wild type conditions, what we are looking at are stomata, and the stomata have these microtubules in them. Also, these are microtubules in live cells. So one of the things that is possible with plants is that we can do in vivo studies. I don't think there'll be too many people happy having glowing microtubules in their neurons walking around. But we can do that with plants. So we can do in vivo studies looking at microtubules under anesthesia and see what happens to microtubules. So, and then we can quantify the microtubules. So this is more observed conditions, but you can see that as we increase the amount of anesthesia, the microtubules depolymerize. And as Stuart mentioned, it's been known for quite a while that anesthetics bind microtubules, and they also have an effect on depolymerizing microtubules. But what's really interesting on this slide is that we screened mutants, and we found a mutant, which is COP1, that does not respond to anesthesia, as the wild type does. And we know this mutant, like I said, really well. It is what is known as an E3 ligase. What this protein does when it's present is that it degrades microtubule-associated proteins, or MAPs. So as we just discussed in the earlier session, that MAPs, microtubule-associated proteins, can stabilize microtubules, and Stuart even mentioned that patients who have stabilized MAPs don't respond to anesthetics as other patients who don't have stabilized microtubules. So this seems to be also true for plants. So this suggests that anesthetics are binding to microtubules and depolymerizing microtubules, and if we can increase the stability of microtubules, then the plants are also more tolerant to anesthetic. So what does that really mean in terms of understanding it from the human perspective? Well, it's quite interesting, because the human and plant alpha-tubulin are 84.47% identical in amino acid sequence. So all the red amino acids are exactly identical between alpha-tubulin in Homo sapiens and alpha-tubulin in Arabidopsis, which is a plant. So there is a very high level of conservation. Why this conservation? Well, let's look at the evolutionary tree, and I think we don't appreciate this enough, that actually plants and animals used to have a common ancestor about 1.6 billion years ago. And I really enjoyed, actually it's the first time I heard Stuart discuss that theory about the origin of life. I quite enjoyed it, because I also think that there is something about the origin of life. And then we know that both plants and animals have mitochondria, so that was the symbiotic relationship of alpha protobacterium. So we both have mitochondria, and then cyanobacteria, which led to plastids or chloroplasts. In fact, if cyanobacteria had also made a symbiotic relationship with animals, we would be green and we'll be doing photosynthesis. So we don't do that because plants do that. So, well, one point I do want to make before we move on there is that this is the disappearance of dinosaurs, and this is where we are today. So this goes back a really long time, and that plant and animals had an ancestor, shared an ancestor. So the conservation of microtubules maybe suggests that those systems pre-exist this bifurcation. So then Stuart and Roger Penrose published this theory of Orch-OR, which led me to think about that if consciousness is the ability to respond to a quantum signal, which obviously like the Bing, but the information is outside, right? Information is outside, and then there's a decoherence and collapse, the moment of Bing. So if you look at, as a photobiologist, it makes perfect sense to me. In photobiology, light is a quantum signal. Light carries information, which is about its color, its direction, its intensity, its duration. How long the light is on, which direction it's coming from, what color it is, all these things matter to plants. So when to flower, when to fruit, bending towards light, all this information is decoded by the plant, by a system that is a biological system, is decoding information that is quanta, right? And then after it is decoded, the whole biological system responds, or what we call photomorphogenesis. It can change its morphology, like I just showed you, which means that the quanta information can have a significant outcome in terms of growth and development. So if that's true, then I think Orch-OR is on the right track towards explaining how we live our lives as conscious beings, and perhaps our consciousness is different, or our experience of consciousness is very different from other living organisms, but there is a degree or some level of ability to respond to the environment and grow and develop in a certain way. So if we accept that, then we can say that light is a quantum information which is perceived by photoreceptors. These photoreceptors are quite well studied, and I've been studying them for past almost 40 years. These photoreceptors can perceive different colors of light, different aspects of light, and we know that these photoreceptors act through microtubules. They don't directly bind to microtubules, but they activate systems that bind to microtubules, and many of them destabilize or restabilize microtubules, which leads to photosynthesis, which is part of energy production. So light leads to photosynthesis, energy production. On the other hand, what I am theorizing is that I call this Aleph. Actually, I published this paper. I will show you the citation in a second. I call this information Aleph. What if there is an information? So light is a quanti, can be quantized, as well as it's a waveform. Similarly, what if there is a quantized information which is extrinsic? We were just having this discussion earlier. And then I coined this term etioreceptor because outside of photoreceptors, growth in dark in plants is called etiolation. So I call this an etioreceptor. Can microtubules be this etioreceptor? They can be. But we don't know exactly what the etioreceptor might be at the moment. But we do know that there is communication through microtubules, and then that may lead to consciousness and development and differentiation. So in essence, what this theory, which I call the theory of spatial relativity, which suggests that space can take different forms, and those forms are relatively different from each other and have different qualities. And then different qualities and properties allow that form to interact with this Aleph or this information, which is extrinsic. And so the experience of consciousness that we have is based on our own properties, whereas the experience of that information a plant would have would be entirely different types of properties, right? So that is this theory of spatial relativity. But it does involve the aromatic rings that Stuart was also talking about. The shikimic acid pathway is the only pathway that makes 3 aromatic amino acids. And one of them, tryptophan, is known to be precursor to dimethyltryptamine, or DMT. And so we know this very well, that photoreceptors inhibit the pathway that leads from tryptophan to DMT and to auxin production. Auxin is really important for plant growth and development. And this same pathway, see if I have that slide, this same pathway exists in human brain. which makes serotonin and melatonin. So in other words, the tryptophan and DMT pathway, which is more active in the absence of light, and when photoreceptors are activated, they inhibit that pathway. So this theory proposes that there is a hidden signal, or just like light, there is some information. Then there is an echoreceptor, and that is involved in activating or promoting this pathway, which would be downstream to DMT. So if you take DMT extrinsically, for example, ayahuasca or dimethyltryptamine by itself, that will activate this pathway. Okay, so finally, I just want to leave with this last slide. So we just discussed that plants have developed processes to produce food for all life on this planet. I think plants are doing that really well. Animals have developed processes to think, rationalize. We think we are more conscious than anyone else. How are we doing with our thinking? Okay, thank you. I'd just like to add, we do have a podcast, so if you're interested, Stuart is on it, and it's called Terra Science. You can subscribe to this. It's on all the channels, YouTube, Apple, everywhere. Be happy to take any questions if there are any. Oh, it was a multicellular organism. The common ancestor was precursor to algae. So it wasn't any more complex than algae, but it was a multicellular organism. It didn't have chloroplast. It had mitochondria, and it was using different substrates to survive. Most likely, still living in some water body.
Two things real quick. One, DMT's been brought up a couple times here. There's differences between certain types of DMT, 5-MeO and then NN-DMT, and they have different implications phenomenologically. So I wonder if that's something you guys consider as you talk about these things, because 5-MeO seems to have a real strong impact on if there is like a ground level of a being experience. So just a comment there. And then a question, would examining non-local effects with light and plant behavior help support this field of inquiry, do you think? So seeing if retrocausal or precognitive, presensory demonstrations would help support a quantum theory with regards to your experiments?
Okay, so first comment, you're absolutely right. There are different isoforms or different forms of DMT, and I think that's easily explainable depending on the affinity and the reactivity to different parts wherever those are. And it will be the same whether it's in plants or animals. Whether they have similar effects, we don't know. Going to the other question, that's a really good question. I've been thinking about designing an experiment that will be kind of non-local type of inquiry. There are many things that we do know that not just non-local but also social behavior. So we talk about social consciousness as well, right? And that exists in plants and trees as too, right? So there are many different types of questions that we can ask there. I was actually talking to Michael Pollan on this podcast not too long ago, and he made a comment which I really liked. He said that we humans are the most conscious, but we will declare AI conscious before we will ever declare plants conscious. I think that's true. We will not consider other lower forms of organisms conscious, and it's the quest is not to declare them conscious, but to explore non-local or these other things to see the different quality of consciousness or different type of consciousness. I think James actually mentioned earlier there are 400 different definitions of consciousness.
Yeah. Over 400, unfortunately.
Yeah. So, if you don't have a definition, then how do we declare something that it is one or not?
You just ask it. Are you conscious? If it falls over with anesthetic, it's, other than the xenon computer guy, yeah, you're good.
Yeah. Okay.
they do have a communication system within other local life, whether it's like sharing water, alerting like of diseases, what have you. Just because they don't self-reflect, because there's no long-form memory, they could still be conscious, just not in the like human form.
Well, I mean, I think we experience consciousness in our way, and we judge everything else that consciousness should be the exact same way. Let's take the example of light again. If we take photosynthesis, you put in one place is the same light. You have different types of plants. They will do photosynthesis at different times, at different ways, because the mechanisms of photosynthesis, even in that, is different. Similarly, over here, we may be conscious, but even humans may be conscious in different ways. So I think we need to think about the spectrum of consciousness, right? So to define consciousness in one way or the other is one thing, which is nothing wrong with that. So, but then maybe there is a spectrum, and what we call consciousness, is it the ability? So I have a definition of consciousness. I'd be happy to share with you. I'm still working on it. But my definition of consciousness is the degree of the ability to respond to the environment with a range of awareness, right? So it's the degree of the ability to respond to the environment with a range of awareness. So you can respond to the environment even without being aware of it. But conscious behavior means we are responding to something or we are reacting to something. That's the Bing moments, the sequence of Bings. And whether those Bing moments, are you aware of it? We are. Plants may not be aware of it, but they have microtubules, like Stuart said, and they have memory. Plants remember if they come across a drought, they will remember it. Maybe it's encoded in the microtubules, like Stuart was explaining. So where do we draw that line, right? It's hard to do that.
Okay, we're going to actually have a panel in 10 minutes when I finish my little presentation, and then we can ask a few more questions and so on. But otherwise, we'll run out of time. So I'm going to do my bit, and then we'll sit up here and we'll take some general questions, if that's okay.